As a reminder, all participants are muted, but if they would like to have a question and ask a question, they can raise their hand or write in the chat or the Q&A. The chat is open for everyone to write their questions. We'll have a Q&A at the end after all the presentations. And yeah, welcome to our second to last session of this webinar series. I leave the floor perhaps to Marie-Noël, who would like to say a few words.
Thank you, Alexandra. Thank you so much. Good morning, ladies and gentlemen. Good morning, good afternoon, good evening. We are here today again together. Today, for today's session, we will listen to our colleague. We have at least four presentations on genomics and challenges related to ploidy crops. So there we go. There we do it through several presentations. So we are so excited to have such a diverse group of learners and professionals joining us at each session. So the session will last 2 hours, around 2 hours, and the Q&A session will be at the end of all the presentation. Please stay up to the end to raise your questions so that the presenter will be happy to share the insight with you. So we encourage you really to be proactive at the end. So thank you again for joining us today. And please, I have the honor to give the floor to Dr. Ranjana, who will start the series of the presentation. Thank you.
Ranjana, you have the floor. We can see your screen correctly. I can confirm. You are muted. I don't know if you're trying to.
Oh, okay. Sorry. I saw that, and then I had to unshare.
No worries. Take your time.
Yeah.
It always doesn't work when you most need it.
Okay.
Great.
Yeah. Good afternoon, everybody, again, and thank you, Alessandra and Madame Marie, for that great introduction. Yes, today we are going to cover a topic which is quite complicated, but this is like a reality with a lot of crops that we have, even the food security crops. So this one is not going down. I don't know what is happening. Go down. No, okay. Anyway, so what I'm going to do is I'm going to give an introduction to the polyploids, like what are polyploids, and then some bits of how we can do ploidy analysis. Because when we don't understand and know the ploidy status of a genotype, then it becomes very difficult when we do hybridization or breeding program. For example, if one plant is triploid, then it becomes sterile. And if you don't know its ploidy status, and then you start, using it in the breeding program, thinking that it might be a diploid, but it's actually a triploid, then you don't get your, progenies. So this is something that is challenging with the polyploids. But also there are a lot of good things about polyploids. So let me just start with that introduction. Okay, okay. So to start with, what are polyploids? So polyploids are actually organisms which have multiple sets of chromosomes, and these are more than the diploid set. So diploid is if maize has, say, for example, 10 as the basic chromosome number, as a diploid, the total number of chromosomes it will have is 2n is equal to 2x, which is equal to 20. That's the diploid number. So that's the normal set of chromosomes. And approximately 50 to 70% of angiosperms, including many croplands, have undergone polyploidy during their evolutionary process. What does that mean? It looks like majority of the angiosperms were actually polyploids. And then slowly during their evolution, they lost that polyploidy and became, due to selection, natural selection or artificial selection, it became a diploid. And there is a big chapter mainly on polyploidy and the evolutionary process and how their diploidization has happened. That's a separate chapter altogether. I have not covered that here. And it is common, polyploidy is quite common in nature and provides a major mechanism for adaptation and as well as for speciation. So if you look at this picture, so this is the normal kind of diploids. So you have mitosis and you have meiosis, and you will have n numbers where you have the spores, etc. It goes through gametophytic situation. Then you get the gametes, two gametes which comes together, fusion, they form a zygote which becomes 2n, and then you have a sporophytic situation, and then the meiosis. This is a kind of cycle that happens in our cells, where cell division happens, and we are all aware of what is mitosis and meiosis. And I will tell you what happens in polyploids. So before I go there, there are how many different types of polyploids are there in this. So based on chromosomal composition, they are divided as euploids and aneuploids. Yeah. So euploids, they constitute the majority of the polyploids that we have, in the angiosperms. Euploids are polyploids with multiples of the complete set of chromosomes specific to a species. So what does that mean? If you have X number or N number, for example, N is 10, you will have multiple of that complete set. It will have 10, 20, 30, 40, something like that. And it will belong to a certain species. I'll come to the aneuploids later. So in the euploids, you can have haploids where you have X number, N becomes X. That means 10. So if a maize has 10, we will say it is a haploid when it is not doubled. So generally chromosomes are always in pairs. So when it pairs, it becomes 2n. Okay. So in this haploid, it is only showing one set of the chromosomes. Then you have diploids, which shows two set of the chromosomes, and that is 2n, which becomes 2x. Polyploids have multiple sets of the same, this, which becomes 2n is equal to 3x, 2n is equal to 4x, something like that. Depending on the composition of the genome, euploids can be further classified as autopolyploids and allopolyploids, and I will explain that in my next slides. So what are autopolyploids? It consists of multiple copies of the basic set chromosomes of the same genome. So that means is the same, not only the same species, but also the same genome. It occurs in nature through union of unreduced gametes. So what happens is the gametes, they don't, they remain unreduced. So they don't break into half. Generally, it should get into half, but it doesn't. The gametes will remain as unreduced. These crops are mainly tetraploids such as alfalfa, peanuts, potatoes, coffee, even the triploid bananas, and their chromosome number can be 2n is equal to 4x, 6x, 8x, etc. So you have, crop, the like potato, banana, peanut, the ploidy is 4n, chromosome number is 48, which means here n is equal to 12. Then you have banana, 3n is 33, it's a triploid. Here n is 11. Peanut is 40, here n is 10. I say 4n again. Sweet potato is 90. So that means here the n is equal to 15. Yeah, so this n means the basic chromosome number. So what happens is when, as I said, they, and the gametes will remain unreduced. So you have, this is the diploid parent where you have, these are in chromosomes are in pair, right? So in the zygote, the chromosomes don't separate. They remain intact like that, and then they get passed in the polyploid offspring. You have now these two becomes 4, and these two also becomes 4. So this is like 4n. Now 2n has become 4n. So what happens is, if you do a karyotype of your parent species, here 2n is equal to 6. So you have 3 pairs of chromosomes here. There is a meiotic error. Nobody knows why this happens. It just happens naturally. So this is something that happens. They just don't divide it into half. The chromosome is supposed to reduce to half to make 2 zygotes. But here, the unreduced gametes will have now 6 chromosomes. So you have 2, because it again becomes, because it is unreduced, so you have these 2 gametes with 6 chromosomes, and here also 6 chromosomes. When they fertilize, now you have 12 of them, right? So this now becomes a autopolyploid because it represents the same genome. And I'll show you in allo how different genomes come together, okay, and can give you polyploids. So this is now your autopolyploid. Yeah, so these can be fertile, because if it is 12, which is an even number, so this can be a kind of fertile. But sometimes when it is 3n, it's a triploid, then it can be infertile. So this is where the complexity comes with the polyploids. How do we know that so one of the plant is fertile, the other one is not fertile? So this is the complexity that we have. Yeah. So haploids are not fertile. Triploids, pentaploids, they are not, fertile, but they can be used in the crossing program. And I think banana breeding will show you how they use it. Okay. So alloploids, they consist of a combination. Now I'm coming to allopolyploids. That was auto. But here they consist of a combination of genomes from different species. Okay. So they result from hybridization of 2 or more genomes followed by chromosome doubling or by the fusion of unreduced gametes between species. So here what happens is, this is more like a interspecies, hybridization that happens. However, the gamete still remains unreduced. This mechanism is also called non-disjunction. Economically important allopolyploids are strawberry, wheat, oat, upland cotton, etc. So here what happens is, you have one species, say for example, it's a diploid and it has got a species A and diploid number is 4. Then you have another species B, which is also a diploid, and its number is 6. And then the meiotic error happens, and then you have unreduced gametes with 4 chromosomes for this A species and 6 gametes from the, 6 chromosomes from the other species. And then when they come together, and this may be normal. You see, they may be reducing into 1, and you can see that it is a normal gamete where 3 have come, right? So now you have a hybrid with 7 chromosomes. 2 that are full and then 3 are just like that, right? And this is unreduced gamete, with 7. So this can also happen. And then this is a fertile hybrid, and the 2n is 10. So this is allopolyploid. And these are also very common, not as common as autopolyploids, but they are also, like wheat is a big example of that. And if you look at the cabbage family, and this is what has happened in case of, this is like, I should start with AA. So this is Brassica nepa. It has N is equal to 10. That is your basic chromosome number, and it has AA. So when it combines with Brassica oleracea, which has N is equal to 9, that means 2N is 18, here 2N is 20, and it has CC, genome. So when these two come together, you now get Brassica napus, which has n is equal to 19 or 2n is equal to 38. Yeah, and it has AACCs, and it's 2n is equal to 4x. So it's a tetraploid. So likewise, you can see from this diagram that you have Brassica carinata, and this is BBCC, and then Brassica juncea and Brassica nigra. So this is how, polyploids are formed. So let me go to the wheat, which is very known. So it comes from an unknown genome, which is BB, which is 2n is equal to 2x is 14. And then when it combines with, Triticum monococcum, which is a diploid species, then you get 2x is equal to 14, which is AB. It is still a diploid. Yeah, because it is 2n is equal to 2x, which is 14, and it is bringing A and B together. Now, these are, you can do chromosome doubling using different chemicals. Colchicine is one of them, but also naturally it can happen. But when you do chromosome doubling, then it becomes AABB, which is a tetraploid wheat, and this is Triticum turgidum or turgidum. Yeah. So when it crosses again with or crossed with Triticum toschai, which is again a diploid, and then this brought a 2n is equal to 3x ABD, and this is 21 chromosome. And by doubling, you got the Triticum aestivum, which has now 2n is equal to 6x, which is a hexaploid with 42 chromosomes, right? So this is how polyploids can be naturally produced, or you through interspecies crossing, or you can also do it artificially, by doubling the genomes. And I will show you one example where this has been done, but at a later stage. Now let me come to aneuploids. What are aneuploids? These are polyploids that consist either an addition, sorry, this is not and, this is n, either an addition or subtraction of one or more chromosome or chromosomes to the total number of chromosomes making up the ploidy of a species. So that means if you have, kind of 2n is equal to 20, then you may have 2n is equal to 18 with a complete subtraction of one of that chromosome. So it can be one or both the chromosomes. So this is how, you can have, these are aneuploids, and these are much more difficult to find out and to work with. Results from the formation of univalence and multivalence during meiosis of euploids. So these are actually euploids, but they only form univalent. Univalent is only one chromosome, or multivalence means more number of chromosomes. And I'll show you in this picture. So there is no mechanism of dividing univalent among daughter cells during. So what happens is, during the anaphase 1 stage of mitosis, they don't divide where they are supposed. Those, univalents don't go to the daughter cells. So this is what happens, and they inherit more genetic material than others. So multivalent, such as homologous, please don't confuse between homologous and homeologous. They are two different terms totally. So here I'm talking about homologous chromosomes, which are a pair of chromosomes. Okay, so multivalent, such as this univalent is one chromosome, and homologous means they are a pair of chromosomes. Yeah, so they fail to separate during meiosis, which is more like unreduced gametes, leading to unequal migration of chromosomes to opposite poles. So what happens is, sorry, this is not mitosis, this is meiosis. So during meiosis, what happens when the chromosomes are supposed to go to the different poles within the cell, they just don't happen. So in allopolyploid, what you see is that the unreduced gametes, but here, even within the cell, the chromosome pair does not separate and go to the opposite pole. So what happens is, when you have two pairs of chromosomes here, they both go to here. So this is, they can go, and this one, they have nothing. So in the first division, you have non-disjunction. Okay, that means they don't separate very well. So this just doesn't have any chromosome in it, while this one has two chromosomes coming together and they again separate, and you may have a haploid gamete, but what you get is trisomic. That means 2n plus 1 chromosome. So you get 1 from here and 2 from here. Similarly, another trisomic. But here you get monosomic coming from here, and another one monosomic, because there's nothing coming from this side. So this one, monosomic, means it has 2n minus 1. That means it should have 2n, but it has only one pair. So this is how aneuploids are. So here you have 2n plus 1, while here 2n minus 1, which is totally different from autopolyploids. So here, if there is a normal disjunction, then you should get in the first meiotic division 2, in your cells. Then what you do is, through normal disjunction, you should, in second meiotic division, you should have 2 gametes with these both. But if again at this stage non-disjunction happens, you get 1 gamete with both and 1 with nothing. Then you cross it with 1 haploid gamete that comes from another parent. Then you get disomic. That means which is normal. That is a normal diploid. Yeah, disomic and diploid are similar. So this is again a disomic, which is similar, but this gives you trisomic and monosomic. So likewise, you can have classification of aneuploids as monosomy, which is 2n minus 1, that is 2n, but it has got only one pair of the chromosome, one chromosome. Nullisomy, where you have 2n minus 2, that means you have nothing. There's nothing, right? So that's what it is. Trisomy, when you have 2n plus 1. Tetrasomy, you can have 2n plus 2. That can also happen. Sometimes you can also see pentasomy, which is 2n plus 3 coming from different this thing. So these kind of chromosomal aberrations does happen naturally. Now, what I was talking about is, you can artificially also induct polyploids. Like, let me come to the induction of haploids, and then producing double haploids. This is something which is very commonly used in barley and many other crops like maize, etc. So when you have n number of your basic chromosome number, that is your haploids, so it can represent your maternal egg cell or paternal microspores or pollens. You know that the pollens are also, haploids. It has got a single, when they come and fuse to the hybridization, they bring the zygote with one chromosome, one chromosome, and the egg cell brings another chromosome. They fuse and they hybridize, and then their 2 pairs are, or 1 pair of chromosome you get. One from the father, one from the mother. So in this case, you can use the anthers, and you can do the culture of anther culture as a tissue culture. Then you regenerate the plants from there. And what this will have n number of chromosomes, or 1 chromosome number. What you do is you use colchicine. Colchicine is a chemical that are used for chromosome doubling, artificially. So you do the colchicine, the chromosome will double, automatically, and then you get this homozygous, completely homozygous, true to type Hordeum vulgare. Yeah. So similarly, you can also take, the egg cell if you want, which is embryo culture. This is when you do the pollen, it is called anther culture. When you do the egg cells, it is called embryo culture. And then when you do the colchicine treatment, you get this, which is also homozygous. Naturally available barley is heterozygous in nature. So this is called producing double haploids, because you are doubling the haploid number to become a diploid. So they are not called diploid, they are called double haploids. How do we do artificially polyploids using colchicine? Like, for example, hexaploid triticale. Yeah, it's a this is interspecific again, but from different species. So this is wheat, kind of, which is a tetraploid. Here the tetraploid wheat has been used, and rye, yeah, which is a diploid, to make a hexaploid triticale. Yeah, so when you cross, and this has AABB genome, and this has RR genome. So when you do the cross, you get ABR. Here the 28 chromosome, that means the N number is 14. No, N number is it 14? Yeah, 14. And here it is 7. So then the chromosome number for ABR becomes 1. Then you double it, and then you get triticale, which is AABBRR. And now it is a hexaploid because it has 2N with 3 chromosomes, and then doubled, then it is 42, which is a hexaploid. Likewise, octaploid triticale has also been produced, where hexaploid wheat was used with AABBDD genome with rye, which is RR, and this is ABDR. And this has chromosome 28 because here it is 21 and here it is 7 coming together 28. Chromosome doubling using colchicine, you get triticale, which is AABBDDR, which is an octaploid. 8, 2N is 8x, and you have 56 chromosomes. So this a pair of chromosomes. So this is how triticale. And now you may say how triticale is better than wheat. You all know that it has brought together the characteristics of both wheat as well as rye. So it is more, disease resistant, more drought resistant. It has got a lot of properties which are much more better than wheat or rye. So this kind of polyploids that you can do artificially, and this has been tried severally, you may be successful, because you may say that, oh, by using colchicine, are all the chromosomes getting doubled? No. So it's not that artificial induction of polyploids is successful all the time. No, it is not. But sometimes, like this one, triticale is a successful case, and it has been produced. It is being grown, and it is being used, etc. Yeah. And now another question you may ask is, oh, can we do it in animals and human beings? In human beings, chromosomal aberrations does happen, and that's why you see some transgender people. But to do artificial induction, it's not possible. It's ethically incorrect. But in animals, these are being tried, but I am not aware if this is being successful. But for, kind of cancer cells, there's a lot of studies that goes through in cancer studies in human beings, mainly because cancerous cells are those which divides what is that called, not in a normal fashion, but in an abnormal fashion, in multiple numbers. And that's where there is an assumption that the cells, they may not become polyploid, but the cell division or cell multiplication undergoes some aberrations, and that's why cancer happens. So ploidy analysis is a big thing in cancer research in human beings. Now, how do you do determination of ploidy? So there are different methods. Like, for example, you can do flow cytometry. There is a flow cytometer. I'll talk about it. And then you can do microscopic analysis. That means you can do the chromosome counting by visualizing the chromosome, which could be very, very difficult depending upon the size of the chromosome and how you can fix it. And then, you also can do DNA content analysis. So you have molecular methods to do that. So it quantifies the DNA in cells. You can visualize the chromosome, physical counting of chromosomes, and this is measuring the total DNA content. That can be done we do it, nowadays using so many different methods when we are doing sequencing, and hopefully the banana or the yam people will cover something about it and tell us how to do that. Advantages, disadvantages, applications, cost. You can at chromosome counting so far seems to be very accurate, but not high throughput. Flow cytometry seems to be one of the best at the moment, but machine calibration and then errors, these also happen. So it all depends upon how you do it. So the ploidy determination, the best way to do it is morphological characters. You can see them morphologically. So you can look at the stomatal size. So A is a diploid, no, sorry, it's a tetraploid stomata, and this is a hexaploid stomata. So you will see that the stomata size is already very large in case of, polyploids. So if I didn't have a diploid picture, but I'm sure diploid stomata is much more smaller than this. For sure, the polyploids are bigger in size, larger in all their morphological characteristics. So if you see, this is a diploid, this is a tetraploid, and this is a hexaploid. So the leaf sizes are also, kind of bigger. This is a diploid seed, and this is a tetraploid seed. This is a diploid flower, this is a tetraploid flower. So morphologically also you can distinguish, but it is tricky because it is also affected by environment and the genotype. So the interaction between the genotype and environment. So if we make a morphological selection that, and say that this is a polyploid, we could be mistaken as well. The next is, which is more commonly used, is flow cytometry and chromosome counting. So flow cytometry means, cyto means cells, metry means measurements, flow means in a stream of fluid. So you let the cells flow in a stream of fluids, and then you measure the number of thousands of cells in it, and then how many, then you start counting and you run it. This is not rapid, it's rapid. Sorry again. So rapid analysis of thousands of cells you can do at a time. So what you need to do is you need to get the root cells in the early morning when the cells are dividing fast. So you bring those roots, then you need to actually fix them in a certain, we call it DAPI, the chemical. After fixing, you run it or fix it, and then you run it through a machine called flow cytometer, and then you get, kind of peaks like this, which you can see in a computer. So what does the flow cytometer does? It measures the cell components as they flow through a fluid suspension across an illuminated path. So that means when the flow, this fluid suspension of cells pass through a light component, then it gives these peaks. And then these peaks are read based on the number of cells that is read at that particular time, passing through that particular illuminated path. It allows qualitative and quantitative analysis of cell components using fresh samples. It has to be always fresh. You can't just bring any sample and at a particular time of the day when the cells are multiplying fast. And then these are the fundamentals is that it measures the cell components using a fluidic system. And there's a huge class that can be given only on the flow cytometry or flow cytometer, how this works. But just to show you, if this is the peak at 50 that you get, you need to calibrate your machine using a reference diploid. Always it is better to calibrate your machine. And remember, when your crop and species changes, you have to calibrate the machine every time. Yeah, you cannot just calibrate once and use it for everything. No, you need to calibrate. So if this is coming at 50, this peak showing it as a kind of 4x that we have determined, or it was determined, and this was generally to begin with, it should be also confirmed with chromosome counting. right? Is this 4x actually 4x? Knowing the basic number of chromosome for that species. So for diploid, the peak will be somewhere in 25 because now it is double, which is 50. And your tetraploid, you should see two peaks, one at 50 and the other one at, no, this is 4x and this is 8x. So octaploid. So this is 100, right? So you need to see at 50, which is a tetraploid, and another peak at 8x, which is at 100. And then you also do the chromosome counting. But counting the chromosomes is so difficult. How can you, your eyes really need to be, supernatural or superhuman, humanly to count all these chromosomes numbers. So this becomes very difficult, but this is most precise if somebody can do it. But this is not high throughput. However, this is very high throughput, but it again depends upon how the machine has been calibrated. And to calibrate, it's better to do with a reference chromosome of basic diploid. Yeah. So you can use genome sequencing in Ploidy studies. Like, for example, you can sequence the entire genome and then you can say, oh, this is diploid, this is polyploid. And you need to, kind of do genomic samples. You have to do next generation sequencing. You have to employ right bioinformatic tools, which are still not that available for polyploids. And my colleagues will tell you, although this is being, kind of worked on, a huge group of people are working on different crops all across the world to figure out different bioinformatic tools. But you also need to cross-validate your findings, yeah, and then integrate it into different advancements. There are other methods that are also available, like molecular as well as karyotyping. Like, for example, you can use your microsatellite markers, which are SSRs, to distinguish the Ploidy levels. It's not that, it works, but it still is difficult. Next generation sequencing, as I said, chromosome counting. There are digital PCRs nowadays. These are very costly, but there are digital PCRs that are being, developed, which can quantify the DNA for Ploidy, and then whole genome amplification also can be used. But there are other methods also, like karyotyping, where you do the chromosome numbers. Yeah, and there are methods like FISH, fluorescent, they use fluorescent, in situ hybridization. This is called not the normal fish you find in water. This is fluorescent in situ hybridization. So it uses fluorescent probes to detect the specific, kind of amount, the DNA sequences and also the chromosome number. So these are mainly used in cancer studies and cancer diagnostics. This is FISH, and there is also another is called GISH. These are still used. There's a GISH is genotyping, I think, based on in situ hybridization. Again, probe based. These are used mainly in human beings for cancer research, but this is also used in plants to some extent, but not as frequently. Challenges in Ploidy analysis technique, as I said, sample integrity. The quality of samples that you do, and then the handling is so important to catch that meiotic division, really to find out how, whether it's a polyploid, because if you can't catch that, it's very difficult. And for aneuploids, it's much more difficult. So majority of the time, aneuploids are done based on chromosome counting because it's so difficult. Data interpretation, like if you have not really calibrated your machine very well, you may be reading the, kind of your data or your samples wrongly. So the peak, whatever you put, the machine will show some peak. Whether this peak is really, corresponding to your samples is something, you have to be careful. Data standardization, majority of the times we may not have that reference, to be used for calibrating the machine for a particular species that you may be working with unknowns. When you work with unknowns and you use another crop. You maybe were, using, say, for example, if you use wheat cells for calibrating your machine for banana samples because you have no known reference in banana, then it is a lot of guesswork that you are doing. Which method you are using, the cost effectiveness, to get the best high quality method. And then there are, technological limitations as well, but this is getting, this is still under progress. Expertise, a lot of time, in doing ploidy analysis, the expertise is very much needed. The people who can really count the cells, or count the number of not cells, sorry, count the number of chromosomes precisely, do the calibration precisely, etc. So this is again, required. Best practices, I think I am talking the same thing: sample preparation, instrument calibration, validation, data validation, and standard controls, controls that we need to use, and then quality assurance. The comparative analysis of ploidy techniques, what you can use: flow cytometry is high speed, accuracy is moderate based on the calibration and catching the right stage of the cell division. Cost is a bit high. Ease of use is also moderate. Microscopy speed is moderate. Accuracy is low, depending upon your expertise in, counting the, what is that called, the chromosome numbers. Cost is moderate. Ease of use is high if your eyes are very good. Genome sequencing, here it says speed is low. I think it is high. The accuracy is high. Everything is high. And then PCR analysis is also like that, depending upon what money you have in your pocket and how you can do, things. This is something that you can do. So thank you very much. I hope I could give a kind of flavor of what are polyploids and how things are, with polyploidy. So maybe, Mary, it's to you. Thank you. Or I should introduce Alessandra, the next.
I mean, sure. I think it was a wonderful presentation. Thank you so much, Ranjana. It was very in-depth. So I will suggest for all participants to start gathering your questions for later. And yeah, we can move to the next speaker. So Agre Paterne, if you're ready, feel free to share your screen.
Is it Agre or it is Julie, or Julie is not around yet?
Oh, no, Julie is here. I saw her earlier. Sorry, apologies. Julie,
no
please
problem.
go ahead.
Yeah. Dr. Julie Sardos is from the Alliance Bioversity CIAT, and she will speak about banana.
Thank you.
Julie, over to you.
Thank you. Okay, good morning, good afternoon, and good evening to all of you. Thank you for attending this webinar. So let me just start by sharing my screen. Okay.
Yes, we can see it. Thanks, Julie. Julie, are you speaking? Because I think we lost your sound.
Julie, we can't hear you. Are you there?
It was working just now.
Yeah.
That's odd.
Julie, can you hear us?
Maybe something is wrong.
Yeah.
Perhaps, Julie, if you could stop sharing your screen and start again and test your audio again. It was just working, so I'm surprised. We can see your screen as well.
Julie, unmute yourself. You are muted. Yeah. Now, can you speak a bit? We
No,
can't
it's
hear
gone.
you.
It's
Yeah.
gone completely. That's crazy.
Yeah.
It was just working.
Maybe then Brigitte can present because she's also speaking on banana.
Yeah,
Just
that's
for the
fine.
time.
And if
Bri-
Julie, you can try and test or leave the meeting and join again, see if that works as well.
Yeah.
And we'll give the floor to Brigitte in the meantime. Thank you.
So Brigitte, Dr. Brigitte Uwimana, she is a scientist here in the International Institute of Tropical Agriculture, IITA, based in Uganda. So Brigitte, over to you.
Yes, Ranjana, let me see if I can get my slides to work. Just read my email.
That's all right.
Go to the window, yeah.
Yes.
Great.
Try to minimize this one.
Yeah.
It's not working.
You can go to presentation mode.
Okay, let's see if it
Perfect.
moves.
Yes, we can see it.
Okay. Good
Thank you.
morning, good afternoon, everyone. Thank you for the opportunity to talk to the big team here. I've been following some presentations, the very diverse attendees, and thank you to the organizers for this platform. I will not give you much details about what happened in banana because it will take a whole day, but I'll give you a flavor of what we do in terms of genotyping. So I will echo a lot of what Ranjana has been talking about, because bananas are not only polyploids, but they're multiploids. What does that mean? In IITA, we focus on cooking bananas. So when I talk about bananas, you try to understand this. So we focused on East African bananas. Those are Matoke and Mshare. Their peculiarities is that they come from the same progenitor, so Musa Kiminata, which is why they're called AA. The Matoke, both of them are not eaten as dessert bananas, but they are cooked when they are raw. So the Matoke, they are triploids. So as Ranjana was saying, 3 copies of each chromosome. The Mshare are only diploids that we work with, but they're the smallest market segment. And in West Africa and Central Africa, the focus is on plantains, which is again a triploid, but with another progenitor coming in, which is called Musa barbiziana. So this is the classic banana breeding night here. But recently we have gone out to the different countries where we operate. What do you actually want us to work on? We were very surprised that yes, the 3 types of banana plantain we work on, they are needed. But people also want dessert and juicy bananas. So dessert bananas are the bananas we know, but in East Africa, they prefer these types of small bananas, what we call, they call them sukarindizi, or in Rwanda and Burundi, I think they call them kamaramasenje. And they also want to juice. When I say juice, also understand beer. So the juice is produced, it can be sold as juice, but in many East African countries, the juice is fermented to produce beer. Those are our future market segments, but for now, we're still focusing on these 3 types of bananas. And it was actually understood that plantains were only wanted in Central and West Africa, but to our surprise, even in Uganda, there's a lot of influx of plantain which come from DRC, from the Democratic Republic of the Congo, and Ugandans would like to have their own plantains. So this is also a future market for East Africa. In West Africa, so we've been focusing on plantains, but there's also a big demand for desert bananas, which could go for even for export or for local market. Now let's dive into banana breeding a bit because this is very important before I understand what we do in genotyping. I said that bananas are multiploids. The bananas we know, even those which are sold as desert bananas, they're triploids. That means they are near infertile, they don't produce seeds. When you try, you can make a lot of crosses. If you're lucky, you get one seed. Something like you can see, let me see if I can get to my pointer here. Something like this. But what we know, like mostly when we get visitors, we show them bananas, they're like, no way, bananas don't have seeds because we're used to this type of bananas. But for breeding, we need seeds. You can't produce a new genotype without making crosses. So what do? We start from 3x by 2x. We cross this. These are mostly can be wild bananas. They can be improved bananas. They can be, as long as they're diploids. Because the diploids, they are mostly fertile. They have a lot of pollen, male and female fertile. So when you cross them, you get all kind of ploidy levels. You get 2x, 3x, and 4x. The majority will be 3x, but what we want actually are tetraploids. Because at this level, when you cross 4x with 2x, you will get a lot of seeds. So this is the pollinated 4x. You can see that it has more seeds than this one or this one. And this one will give you a triploid. So you can see the combination of the chromosomes at meiosis, which produces a reduced gamete or not reduced gamete to produce a certain ploidy level. What you don't see here is that this 3x will come also with other 2x and 4x's. But this 3x is what you want because it will go back to seedlessness. So you don't want to give farmers hybrids with seeds. And it will also combine, it will carry some level of quality from the triploid and resistance from the diploid with increased resistance from these diploids. But what you see here is only two events of meiosis, which is a very limiting factor in banana breeding. So what do? We wanted to develop these parents independently. So we have a 2x by 2x improvement program, 4x by 4x improvement program, then also chromosome doubling, as Ranjana was talking about. We give you more tetraploids, which can also be included in this program to produce the improved tetraploids and improved diploids that you need. Now you understand this complexity that we have to navigate at every level. So what are genotyping challenges? First of all, as I said, it's a current propagated crop. You get very limited recombinations. To get your population will take you, like you can make your crosses for 2 years to get the population that you wanted to work with. The addresses and they are triploid and they are sterile. So the seed set is very poor. And also, in addition to that, germination is also very poor. We need to germinate the seeds under tissue culture, so we extract the embryos and they culture them under tissue culture conditions. The propagation is very slow. So in the field, you can get out of one plant, you can get only 5 plantlets. So again, with tissue culture to be able to get your phenotyping population in the field with the required replications. I talked about multiploidy. When you make one cross, you can get at least 2 ploidy levels and sometimes 3 ploidy levels. When I'm talking about ploid analysis, this is our routine work. You can't have a banana breeding program without a running ploid analyzer. It's expensive, it's time consuming, but you have to do it. There's no choice. In terms of genotyping now, because of the polyploidy level, the analysis is not straightforward. So I envy other crops that send their samples out, they get their result out, they go ahead, what they are done. We have to go additional steps to get our right genotypic data. It's also outcrossing, which means it's higher heterozygous. It's not straightforward that you're going to go from F1, F2, F3, the recombinant inbred lines. You need to target, depending on the population you're looking for, and the heterozygosity in each parent of them. You will target the number of markers you want to be segregating, depending on the parents that you're crossing. As I said, we get to more than one ploidy level. So now what do you do with the analysis? You either reduce your population to stick to one ploidy level for your, let's say, for your QTL analysis, or you need to be inventive and find other ways that you can combine your ploidy levels. This is where we are today. And again, genotyping is a game between resources and accuracy. If everything was perfect, we would do 40x sequencing of everything and get everything perfect, right? But life does not come that way. Resources are limited. So we have to do with the standard genotyping platforms and still get the data that we need. This has been our journey, at least my journey in banana breeding as a molecular breeder. When I joined, GBS was the top of the genotyping platforms, but calling by the dosage in GBS was, the GBS itself was coming with a lot of missing data. Now calling by allele dosage is another nightmare. You can try with JTK, but because of the low sequencing depth, the accuracy is very, very poor. You end up with some genotypes which are missing or which are miscalled. There was a big promise of OMSIC, which Sweet Potato was using. We tried it. It did not work for us. We ended up with missing a lot of genotypes, with more than 40% of genotypes missing to be able to get the right balance between a good coverage in markers and a good coverage, like the right number of the genotypes. You would lose on both ends. We are now doing that seq, but that seq is still not good for polyploids because you still can't call by the dosage without going into inaccuracy in coding. So we have come up with another system of coding, which we call continuous mapping, where we call allele frequencies. In this system, you don't call the genotypes, but you go to the level before calling the genotypes and look at the allele dosages of the reference to the alternative allele, and you correct for the differences in the sequencing depth for each genotype, for each data point. So this is what we're applying now. I will tell a bit about CASP. CASP markers, again, you will get the results as diploids, but we need the result as polyploids, triploids, tetraploids. So after I get the result, to apply fit poly to call by the dosage. We are moving to that tag, which will be very helpful for genomic selection, not only as repetitive markers that you can keep on adding more data sets on, but also for calling by the dosage for this multiploid crop. So this is a summary of what we do. Different purposes gives you a different genotyping platform. For diversity studies, we go for that seq. There, we don't care about the ploidy level, we just want to know the relationship between the different genotypes. For trait discovery, we want to go as detailed as possible, but we need a lot of markers. So that's why we need now to balance accuracy and resources. So we go for that seq. But when we're dealing with triploids, we move to continuous mapping. For once I've fixed our markers in the QTL analysis, and they want to move to markers we can use now in the marker selection, the markers are converted to CAS markers. And as I said, for the polyploids, we add allele coding by allele dosage. I'm coding using fit poly to add the dosage level. For genomic selection, as I said, we are moving to that tag. And for quality and quality control, This is mostly used for cross-verification. It's also used to, let's say you have a hybrid which has been selected at a certain stage. You move it from one evaluation stage to the next one. You have to go through tissue class to multiply. A lot can happen. So those genotypes, especially those which are promising genotypes, they're always tracked using a very few cast markers in the QC. They're highly repeatable, which means we can just have the reference, the fingerprints of the genotypes that we need, and we keep repeating, analyzing the new genotypes or the new plants to make sure that we are still having the same identity using cast markers. That's what I have for you today. Thank you very much.
Thank you so much, Brigitte, giving an overview of, the difficulties or challenges, but also opportunities, in a crop like banana, which is a food security, banana and plantains, which is more like a food security crop in Africa. So, Julie, are you ready now with your
hopefully.
Sure, we can hear you. We can hear you.
I don't know what happened. I'm sorry. I had to restart my computer completely. Nothing was working anymore. So I had no possibility.
Yeah. So please go ahead, Julie, with your presentation.
Okay.
Yes.
So let me share my screen. Okay. Okay, so
Yes.
I will also discuss about banana, but here we will see how we can make Ploidy inference from high-throughput genotyping data. So this presentation is a follow-up of the presentation I did last week on the high-throughput genotyping data and the VCF format. And it will be structured in 4 parts. It will be a short presentation. So first, I will give a little bit of context on banana. Well, Brigitte already did it, but I will give a small context, but also on the work we are doing on banana. I will make a small refresher on 2 concepts that we saw last week that are allele depth, so AD, and site depth, DP. And then we will see how we can infer Ploidy from these 2 measures. So we'll see the principles. I will show you a tool, GBiasDiv, for which you don't have to have coding skill to use, and then we will see examples. And at the end, we will see shortly how important it is to have high quality data to make this kind of inference. So first thing, banana. So I assume everybody knows the fruits, but maybe you don't know the genomic of banana. You don't know where's the crop. So the basic number of chromosomes is 11. Wild bananas are diploid and they are fruits full of seeds, but cultivated bananas are fleshy seedless fruits. So the crop is vegetatively propagated. Banana originate in a wide region in Southeast Asia and West Oceania, and all the cultivated bananas existing nowadays are hybrids between several ancestors with a minimum of 3 and a maximum of 6 or 7. And as previously attested, cultivated banana can be either diploid, triploid, or tetraploid. So as I told you last week, I work for the Alliance Bioversity International and I work for the Banana Genebank. So our collection is named the International Transit Center, ITC, and it's based in Belgium. So our accessions are conserved there in vitro. So we are in a university, we have a lab, we have access to flow cytometry, so we can measure the ploidy of our different banana accessions. But I told you this last week, but the tissue culture can induce aneuploidy, artificial aneuploidy. That is to say that during the process of multiplication, chromosomes can be lost or they can duplicate. And this affects the genetic integrity of our material. So for us, it's very important that we are able to monitor this risk. We have also another part of our work that is dedicated to on-farm diversity assessments, and we also support national collections in characterizing their material. So in these two cases, most of the time we don't have the possibility to perform flow cytometry with this material. So Ploidy can be assessed only on the morphology of the crop, but first of all, it's not always possible if the plant is not at the right stage, for example, but also you need a certain level of expertise. So as Dr. Ranjana was saying earlier, it's not always accurate to do this on morphology. So now a little refresher on these two concepts that we saw last week, allele depth and site depth. So if you remember well, I told you when you call your genotype from your sequencing experiment, the reads are mapped on the reference genome. And then you have a special software that is called the caller that will check all the reads specifically for variants. And then it will count the number of reads at all the different variants on the different SNP position. So for example, on position 1, you have 11 reads that are with the reference allele A, while you have 7 reads that hold the alternate allele T. So this information is kept in the VCF, so your data file, under two measures. The first one is AD, allele depth. So this corresponds to the number of reads for each allele. So for example, here you have two numbers for allele depth always. The first one corresponds to the number of reads for the reference allele, while the second number, here it's 12, correspond to the number of reads that were counted for the alternate allele. Another measure that is given is in green. It's DP, the site depth, and corresponds to the total number of reads that were observed for the given SNP or site. So now, how can we use these measures to implement Ploidy? So last week we saw that the number of reads, so DP on the VCF file, was correlated with the relative quantity of DNA in your sample. So we identified a duplication in a diploid, for example. But in fact, this principle can also be applied to allele depth. So that's the allelic dosage that Brigitte was mentioning in her presentation. So that means that the number of reads that are detected for each allele will be correlated to the number of copies of each allele that will be present in your sample. So here, let's take the example of a diploid sample. So you look at the heterozygous sites. For diploid, you will have one haplotype that will bear each of the allele. So there will be a similar amount of DNA for both alleles. So this will result in an equivalent number of reads for each of these alleles. And so the values of the two AD measures should be similar. Therefore, if you make a very simple calculation, so you take AD, the number of reads for the reference allele, and you divide it by DP, and you do the same for the alternate allele, you will end up with numbers that are around 0.5 that correspond to half of the total reads for each of the allele. Now, if you have a triploid sample, you have 3 chromosomes. And if you have an heterozygous site, you will have an unbalanced number of copies for each of the allele. So one allele will be present in one haplotype, while the other allele will be present in 2 haplotypes. This will result in an unbalanced number of reads for the different alleles. So if you take AD for the reference and AD for the alternate, and each of these measures are divided by DP, the total number of reads, you will end up with results that will be around 0.33 and 0.66 respectively, and that will correspond to 1/3 and 2/3 of the reads for each allele respectively. This can also be applied to tetraploid samples, but there you will have 2 possibilities for heterozygous sites. In the first possibility here, you see that there is an unbalanced number of copies for each of the allele. So one allele is present once, but the other one is present 3 times. This will result in an unbalanced number of reads. And so if you go back to this calculation of the ratio AD divided by DP, so the allele depth divided by the total number of reads for the site, you will end up with 2 values. The first one, that will be around 0.25. And the second one that will be around 0.75. And that will correspond to 1/4 and 3/4 for each of the alleles. The other possibility for tetraploid is that you have 2 copies of each allele in the genome. So in that case, you will end up with a balanced number of reads. And at the end, if you make the calculation of the ratio AD divided by DP, you will end up again with value, like in the diploids, with value around 0.5 that will correspond to half of the reads for each of the alleles. So it's possible to use these measurements to check or infer ploidy for the sample for which you have a doubt or if you want to check things. And this, if you go to the CIAT website, you can download a software that is very useful and for which you don't need to have any coding skill, that is named GBSDiv. So in GBSDiv, you can input your VCF file. So you have to make to tick specific boxes, then you run the analysis and it will treat all the sites of your, in your VCF, all the data of your VCF in some minutes, and you will be able to compile some statistics. And among the statistics, you will have distribution plots for the AD by DP ratios. So here is an example. I will explain to you the graph. So each color line corresponds to a chromosome. So in banana, we have 11 chromosomes, so they are all, so all the lines are different chromosomes. I just forgot to take off the mitochondrion from my analysis, so it's there, but it's not a problem. So the graph, the x-axis, will show you AD divided by DP for the dominant allele, that is to say the allele presenting the highest number of reads, while the y-axis will show you the number of SNPs that were observed for the specific value. So here is in percentage. Of course, there was not less than 10 SNP in my analysis. So here you check the distribution of the AD by DP value, and you will see that it is centered around 0.5. This corresponds to half of the reads for each allele. So this is the diploid situation. Now, we have another plot, so it's still the same. On the x-axis, it's still AD by DP divided by DP for the dominant allele, the number of SNPs on the y-axis. And you can check that for all the chromosomes, the distribution has a different shape and is centered around 0.66. So this corresponds to 2/3. So in fact, here we are in a situation where we have 2/3 of the reads for the dominant allele and 1/3 for the minor allele. So here we have a triploid profile. Then we have also, we can have this kind of peak. So here you can see that the shape of the curve is a bit different. You have a peak, a first one around 0.5. So this suggests that there's an equivalent number of reads for each of the allele. But there's another peak in around 0.75, corresponding to 3/4. So here we are in the situation where in some heterozygous sites, you have a balanced number of reads between the two alleles present. And in some other heterozygous sites, you have an unbalanced number of reads for each of the allele, with the dominant allele representing, 3/4 of the reads, and the other one 1/4 of the reads. So here we have a tetraploid profile. Now, there's also an interest to have the data disaggregated by chromosome, because here we have a distribution that shows that 10 of the chromosomes have a distribution around 0.66, so this corresponds to a triploid. But we have chromosome 6 here in light blue that has a different shape, and it is centered around 0.5. So here we suspect that one copy of our chromosome 6 is missing for this specific accession. So here I just put on the same slide all the different examples that I just showed you for you to really realize how different those shapes are. So it is very powerful, and it really helps when you need to check ploidy for your samples. Now the importance of data quality, especially the coverage and the SNP density. So in fact, the clarity of these plots depends really on the quality of the data. So you need to have sufficient number of SNP, but also sufficient number of reads per sites. From our experience, plots looks better with RADseq than with GBIS. We haven't tested other methodology for cultivated bananas. This is linked to the methodology, the lab work in RADseq. The protocol is different because you have a sharing steps in which DNA fragment of equivalent size are selected, and this step does not exist in GBIS. And also you will see in the next slide that when sequencing coverage is low, it is more difficult to interpret the results. So, for example, on the left, you have a plot that I made from low coverage GBIS. And you can see that the shape of the curves is very difficult. I mean, it's very, very difficult to interpret. I would not be sure that it's a diploid or a triploid in that case. On the right, you have an example with GBIS also, but we had a problem at the lab. It was a high density sequencing, but there have been a problem during library construction and partial enzymatic digestion. So in fact, here again, I'm not really sure what the ploidy is. So those two samples were both from on-farm materials. So flow cytometry was not possible. So now we have to investigate other means based on alleles and genetic study to assess their ploidy if we can. So as a conclusion, I will tell you that this type of analysis is not banana exclusive. The software GBIS div can be used with any species, and it allows many different things. So the first one is to compile basic statistics about your data set, such as missing data or the rate of heterozygotes. It can also help you to infer ploidy for your samples. But also, and I didn't have time to show it, but if you want to calculate pairwise genetic distance in your data set, well, it can make it taking into account ploidy. There was a question during the GWAS session about how to correct for kingship when you have a polyploid and you want to perform GWAS. Well, in fact, it's possible to calculate pairwise genetic distance that will take into account ploidy up to tetraploid. And you can even have mixed ploidy in your sample and it will work too. So it is a tool that is quite easy to use. It's free, of course. You can download it in CIAT's website. And you don't require any coding skills. So it is really powerful and helpful for your analysis. So this is it for me, and I really thank you for your attention. You can ask your question at the end of the session if you have some. Thank you.
Thank you so much, Julie. This was really good, and you know that Ploidy and then doing analysis using all those it's not easy, but bioinformatics is getting better, and people are really working hard, because these are food security crops, and we need to make strides in that. And with two bananas that you have seen, I'll switch now to yams, which is the Dioscorea species. And we have our scientist, Dr. Paterne Agre, who has been working on yams. And this is, again, a very, very multiploid crop with a lot of challenges. So over to you, Agre, and he's from IITA as well. Thank you.
Okay, thanks so much. Let me try to share my screen. So I think, yep. Okay, can you see my screen? I think yes. Let me see. Should be able to see. Can you see my screen?
Yes, but which is more like a,
Oh,
are you showing us the data?
no, sorry. I think I'm sharing another screen. Okay, yeah. Okay.
Yeah, we can see it.
Yeah.
What's happened here? Let me see. Minimize.
It is perfect, Agre. We are seeing it perfect.
Okay, perfect. I don't want this one to show. Okay, so I will be presenting on genotyping and ploidy analysis in a vegetatively propagated crop, the case study of yam. So this will be my outline, just a brief introduction. Sequencing platform we have been using in the yam breeding program. Germplasm management.
Agre, you're not sharing.
Agre, yeah, you are not sharing. Your
sharing.
screen is not sharing.
Okay, a minute. What of now?
No, not yet. Yeah, yes, now, but put it on presentation mode. Yes, now it's okay.
Okay.
Go ahead, Agre, yeah.
Yeah, so I was saying that I will just, my introduction will be a brief introduction on YAM sequencing platform we have been using in the breeding program, germplasm management, the quality control we have developed, how we come up about it, and then the effects we are seeing on the polyploid, mid-density SNPs markers, and then for what purposes we are using in the breeding program. Brief information on the QTL mapping, and then how we are dealing with the Ploidy, and then I will just briefly talk about the pangenome, how we are using it in the breeding program, some high throughput phenotyping, and then the YAM genome hub that we recently developed. So yam is not only a food crop, it's also considered as a cash crop because it's a very expensive, and then feed a lot of people across the sub-Sahara. It's also a social cultural crop whereby I think it's the only crop that have a festival date in West Africa. We have a lot of species, which is good, which is also not good, more than 600 species. And then we have 11 cultivated and semi-cultivated. The genome size is estimated to be around 500 to 800 MB. It's not so heavy like wheat or other genome. It's a polyploid crop, and also the level of the heterozygosity is very high, which make the genome very complex during the analysis, and then we'll talk about it during the. So these are the different food that's kind of like generally originated from the yam. You can see some pounded yam, some roasted with fire fried, and then recently ice cream developed from the yam. So for the platform we have used, and then we just, in the breeding program, we are focusing mainly on two species, Alata and Rotundata, but we have some intermediate species that we use them during the intro aggression. So my presentation will be mainly focused on the Rotundata and Alata. So we have used whole genome resequencing, whereby we have 332 genotype, both breeding lines, some landraces market variety. We have some breeding panel whereby we use high density the diversity array. We have used before GBS on 206, which is for mapping population, and then we use that one for QTL mapping and to develop our genetics map. And then from all the previous genotyping we have done, we use those information to develop a quality control, quality assurance markers, and then we use them for routine activity in the breeding program. We have genotype, more than 10,000 genotype, especially focusing on early generation using the mid-density data that we develop. So we show how we develop those ones. And then we have the mid tag that we are using for genomic selection and then for GPCP also. And we have right now 6 genotype sequence using a high file and then high C combined with PacBio and long read sequencing, including like more than 1,000 also genotype that we sequence with high depth long read Illumina sequencing. So if you look at this graph, we have collected a lot of material from West Africa and also East Africa, whereby we have Rotundata, Cote d'Ivoire, Ghana, Uganda, Benin, and then also we have Nigeria. So we sequence this material using different platform, and then we use some algorithm to select common markers. And then from here now you can see that we have, in terms of species, we have a well-classified group of variety. We have Rotundata here, we have some Alata, and then we have some Princelis. Then we have a mixture of some Bobifera and then some widely related species here. The same thing also where we use the network analysis. But however, if you look at the country, you could see directly that we could not classify the material-based country, which is normal due to exchange of material between a neighboring country and also the breeding a program established in West Africa. And then in the breeding program, sometimes you are dealing with a lot of material, but those material you may be facing some synonyms, homonyms. So it's good to be able to clarify them to know exactly how many unique material you have in your breeding program before you could use them as a progenitor to establish your breeding program. So in the case of yam, we use during the sequencing, we intentionally integrated some accession, the same name, but we hide the name to be able to, when we receive the sequencing information, we should be able to use them to identify at what rate can we declare some genotype as duplicate. So for the case of yam, we end up at 0.04 between two samples. We can declare them that they are duplicate. So using that information now, we're able now to identify all from the 332 whole genome resequencing 173 that are unique. And then we use this information now to develop now what we call a iterative group, what we are using currently in the breeding program to develop now different product, to develop different product profile based on our market segment. Still in the exemplars management, we also make use of DAPC because we have a lot of analysis, but some of them are applied for yam, while some of them are not. So we make use of DAPC, and then we can see we have a very good classification. Neighbor joining also based on identity matrix also work very well for the case of yam. And then this is kind of like very good information for the either for the polyploid group So this one, what happened here now, we have a lot of material, and then we want to see if the heterozygosity level have effect on the classification method. So you can see when we select marker with low heterozygosity level, you can see that you cannot really classify your population. Why, if you select marker with high heterozygosity, you can see that this marker will be able now easily to classify your population. So this also have an effect when you want to use such marker for association mapping, where I will talk about it later, how this information affect the trait length analysis, and then how in the breeding program we're able to overcome this issue. This work has been published, and then more information is available. You can read it also. Now for the quality control, I remember when we started this work was also very difficult. We selected almost twice markers, but we always failed to have the right marker to use for quality control. So for here now, these are kind of like the different information we consider to select the high polymorphic information markers that could be used for quality control. We focus on balance observed and then expected heterozygosity, minor allele frequency. Then we focus on the predictive parallel. The GC content is also very, very useful to not select marker with high GC content. And then here now, if you focus based on the putative parallel in your population, when it's above 1.5, you know directly you are in the case of a polyploid, which is our case here. So running this analysis also gives you some basic information that to know the kind of population you are dealing with. Is an R-based program that which can be easily used and then is available. And then using this information, we focus on two methods. One is based on purity, and then another one is based on simulated. We're able to select 100 markers, because you can see here now, selecting with 50 markers, you already predict almost 80% of the total information. So using the two methods, we selected 50 markers from the each method and combine all of them. We end up with 100 markers. And then those 100 markers now were converted into a competitive alleles marker that we are using in the breeding program for pedigree verification, variety tracking, and then for yam species identification. And then you can see we use this marker now in the breeding program, and then we're able now to classify all the yam species we have in the breeding program. So rather than to use a million or thousand of SNPs markers, we can just use these few markers to do your routine activity in the breeding program. And then this also, as I was saying before, we are using also these markers that we develop in the breeding program to, in case you make a crossing, we just select few markers and then we check the progeny, and then we could easily identify the non-hybrid based on the allele segregation pattern. So this also, you have more information in this paper. You can read about it, how we use it also. Now for the mid-density development in the breeding program, as I was saying before, we have thousands of markers, but for genomic selection, we don't really need a lot of markers to be able to achieve high accuracy. So in the yam breeding program, we have put in place a well-developed program and then a pipeline whereby we can use it to identify high polymorphic information markers that can be used. So this is the initial marker where we have, I think this should be around almost 14 million SNPs markers. And then the first thing we did, we proceed with the linkage disequilibrium analysis to see which of the markers are in high linkage. As we define some window, and then we remove those markers that are highly correlated. And then from that, we're able now to narrow down the number of the markers to 3,000. And then you can see how the 3,000 SNPs markers are distributed. What we did also, we are using this information not only for genomic selection, but we included the quality control markers and then the trait length marker that we identify. We included them into this panel. So whenever we are genotyping our material, that means we can subset this genotype and look at some of the progeny or the population, which of the the progeny have the allele of interest. So this set of markers is very useful for the breeding program as it carried a lot of information. It is also available on the EIB platform. If you are working on YAM, you can just download this one and use the marker for your genotyping. Now, this is the QTL now, how we are doing this one in the breeding program. It's not easy if you see what Banana and also what Ranjana has presented. To deal with the ploidy is not an easy task. So for the case of the yam, this is a latter where we started, we made a crossing between these two parents. So to be able to achieve this one, we use a tool developed by the Polyploid group, what we call a map poly is an array package. So the way we proceed is just to take the VCF information and then you provide the two parents. Then we scan the alleles across the two parents and then the progeny. It should be able now to predict the ploidy level for you. So for this case, you can easily see that the population we are dealing with here is a diploid information. Screen, okay. Yeah. Okay, sorry, I was looking for, okay, pointer option. Okay, yes. So for this one, you can easily see it here that the ploidy level is 2, which means we are dealing with a diploid population here. Of course, this is a prediction, and then you can also check the prediction accuracy. So this package also will give you the presence of the missing information, the number of markers. And then another test also we did also, you can see in this case whereby we have different information. This one now, when we are in the presence of population of hexaploidy, you can see the level of the ploidy is 6, and then you can see how the alleles vary from the two parents. More information is also available in this paper for if you want to see how we did all this analysis. And then using this one, you can see you have your set of markers, and then the package will be able now to tell you the number of the marker is removed, and then the number of the genotype the number of the marker distorted, and then the number of the genotype that not following the correct segregation. So you do all those quality control before you start your analysis. From there, we reconstruct our pairwise recombination, which give us a very nice information across the 20 chromosome. Of course, we can see some artifact here, which we correct before we finalize the construction of the genetics map. Using that information, we're able to construct already a genetics map for Rotunata, what we are using in the breeding program, and then we have for Alata also what we are using in the breeding program. So for the QTL analysis, we use the QTL2, RQTL2, and then we're able to identify some QTL related to the disease, which were phenotyped using 2 methods. Plants in the field and then detached leaf as assay. So we have 2 papers on this one. One is from Dr. Ranjana and then myself also. And then we put these are the two three chromosomes that kind of like have the different QTL we identified to be associated with this disease. Another thing also we did for the trait lengths mapping, which is very key, so that you not identify some markers but you cannot use it in the breeding program. So as I was saying before, we wanted to see how the heterozygosity level affect the association. And then you can see here now we have our raw read, and then we use the GATK. The GATK also have that ability also to predict your ploidy. You can change the pipeline. And then what we did after identify our VCF, now we subset the material. And then by subsetting this one now, we divided this one into different kinship based on the heterozygosity level, which were used as now covariates in the model. So using this one now, you can see if we are using marker with low heterozygosity, you can see the expected and then the observed, they are not really matching. But using marker with high heterozygosity, you can see that we have a very good prediction both with the observed and then the expected. So we now concluded that this information is very good for at least a crop like yam. I think this also can be replicated for other crops that are having having issue on ploidy and then complexity. We use also network to kind of see, okay, which of those markers are common across all the model we use, and then we focus on those common ones to replicate in the breeding program. These are the list of the markers that we have validated some, and then we are using them in the breeding program. In terms of marker deployment in the breeding program, this is for plant cells, and then early generation, we have almost 4,000. And then we, based on this information we have on the male, female, at early generation, we can predict to, I think, almost 90%, or yeah, 80, 70, which of them are male, which of them are female, because the sex in yam is very challenging. For the first year, second year, you are not going to see sex on yam, which will not allow you to make a very good selection in the breeding program. So we have also the marker, but we are having some issue. You have a marker that at some time, if the prediction is not very accurate when you are within male and also within female. So this one now will now assist us to kind of start thinking out of the box, where we started doing the pan genome and also some transcriptomics, what I will briefly talk about it later. This is for the disease. For the disease, we have now a very good markers that we are using in the breeding program. But of course, in the population we use, the accuracy is very high. But when you are moving this marker from one population to another one, the accuracy starts decreasing, which we are suspecting based on some presence of transcriptome transposable element. What you will see how this one affect the yam genome. This is still for the disease, and then as I was saying, the two marker we identify, if we look at the two marker, both of them, they predict at 81%, which is also very good. So we can use either one marker or another one to do our prediction. Now, the first reference genome that has been developed, we compare it using the dot plot analysis in the breeding program, and then you can see that the second one have more information in terms of length and then coverage. So, which is what we are expecting. Reference genome should be always improving against the previous one. Now, why are we doing a reference pan genome now in yam? The previous reference genome I just presented, they were all developed based on linear regression, which means we are missing a lot of information. And then some genetics variation are totally missing because it doesn't represent the entire genome or the entire diversity you have in your core breeding. You can see here, if this is one of the reference genome. If you are using this one to do a mapping of this one, see you will be missing this chunk of information here. But by combining all this one, you are sure that, okay, any alignment you do using this combination, that you should be able to get a very accurate reference genome. So that is what led us to the development of the pan genome. how we identify this material we use. So we did some pedigree analysis to see which of the progenitor has contributed to the development of progeny in the breeding program. So we identified those progenitor with high cycle, which is kind of like the level of contribution. So these are kind of like the parent with high contribution in the breeding program. So we selected them and then we sent it. We used two approaches for sequencing. We sequence them using a hi-fi, and then also we use a long read combined with a high-C and then PacBio. So all those information were embedded during the analysis. And then using this one now, you can see for the pond genome, for the genome scope, you can see that we have 98% of genome coverage, which is kind of like very good. Of course, it's still with some crop we are expecting for 99%, but for yam, which is a monocot, is really as acceptable. So the Busco summary here now, if you look at the 6 genotype, we have sequence using the pond genome approach and then the genome scope. You can see that we are 92, 93% genome assembly accuracy. And then as I was saying before, this is really acceptable for a monocot like yam. And then the number of the gene also is very high, which is okay. And then improved version is good. And then the genome, the scaffold 50, and then the genome, the scaffold 50. So I didn't put the scaffold 90. So we're able now to estimate the genome size, which is also within the range of what we are expecting for rotundata and alata. And then in the case of the polyploid crop also, you may be seeing some very missing portion of the genome here, which will start giving you a lot of headache during the assembly. So you have now to start going by what could be the reason. Is it sequencing error or what really happened? So for this case now, what we did now was kind of like the alignment we did was not properly. So we have to change the entire program and then use another tool which helps us now to assemble all the haplotype here now. So you can see this is chromosome 1, but inside chromosome 1, we have two haplotype 1 and then haplotype 2. And then we compare them later to see if we have some variation between the two haplotype. Of course, some may show some variation, while some of the haplotype may be identical. So for that, you can decide to use the haplotype reference genome for your alignment, or you can use the entire reference genome without considering the haplotype. And then, as I was saying before, you can see here now we have done a proper alignment for this genome, and then it's estimated to be around 530 MB. And then we can see some variation on around chromosome 6, chromosome 19, and then chromosome 20 here, which we are suspecting them to be related to the sex information. More analysis are still on their way to be able to clarify what those variations we are seeing on chromosome 9, chromosome 6, chromosome 11, 19, and 6. So we should be able now to elucidate those variations we are seeing on those ones. Then still on the pan genome now, what we did, we used two approaches. We have the pan genome based, the KMA analysis, whereby we have the two species, rotundata and alata. And then you can see here now the pan genome, we're able now to classify the two species. We have the alata species here, and then we have the rotundata species. So the pan genome approach based KMA, you can use this one to really classify different species, and then it will also allow you to see your source of variation within species and then among the species. Then when I say this is Rhodonata, and then what we did now, we look at the haplotype, and then as I was saying, you can see that if you look at the haplotype A and B, no more variation for this one. So during alignment, if you want to use haplotype B, or you can use haplotype B or haplotype A, you are not going to see much of variation. The same thing also apply for Alata, and then you can see also that we have some variation among the haplotype A and B within some of those species and then within variety. So this one is very useful information when considering the pan-genome approaches. You also try the PGG approach also, and then you can see with this information now, you are sure that you are in the presence of a ploidy information, because some of the portion are totally missing from one genotype to another one, which means definitely one of the genotype may be carrying more information, more information than one of the genotype present in the diversity panel we use. So here is still really challenges for us because the number of material we use is just 6. So we are adding more information now, 41, covering different species, different crossing, different progeny, to be able now to clarify this information we are seeing in the pan genome, to be able to clarify also how the heterozygosity, the ploidy affect the pan genome result, and then how to embedded all those information into a single reference genome to be used in the breeding program. Then looking at Alata and Rhodonata, you can see that for the number of the gene we identify is around 41,000 for the gene for the total gene family. The private gene is around 37% when we compare it with all the monocot, which means the yam still have a lot of kind of like specific or private gene that are only available for yam, which could be explored for future discovery for disease or for some acclimate related. So we are doing kind of like gene annotation and refinement of all these private gene to be able to come up with more information. And then when you look at Alata and Rhodonata here directly, you can see that the genome are totally different. This may not display it very well, but you can see here now, if you compare Alata, they only have 26% of the genome that are kind of like are maintained when you are focusing on the two genotypes. So as I was saying here now, if you look at this one, if you look at Guagua, which is Rhodonata, and then this one is Alata, and then you can see that we have some chromosomes that are kind of like that are sharing the same information, while you see some chromosomes that are totally different. From chromosome 7, you can see that chromosome 7 in Rhodonata have some part of the genome in chromosome 12. So the same thing also on chromosome 12. You can also see some inversion of the region, like if you see chromosome 15, you can see for the A segment here is a segment B in Alata. So all those information, we have them already in the breeding program. So which really help us when we are doing our alignment and then making a very good decision, and then selecting of marker that could be also very useful from crop for across species, not developing marker for Rhodonata and Alata. And then the transposable element also are very useful, and then it's very good when you have a polyploid crop to know how those transposable elements, the proportion of them in the program is good to have a very good information on them, and then how those ones could be used to kind of like avoid some misleading information. So for the case of yam, you can see that we have done the T analysis, and then we're able to identify the proportion them of across each haplotype, and then we have mapped them across all the haplotype of the 20 chromosome. Since we have haplotype A and B here now, we have 40 different haplotype in total. We are also developing some some high throughput phenotyping in the breeding program for quality analysis. And then we have Visio that will be implemented, some NIS, which will help us to kind of do some marker validation. High throughput phenotyping, and then we are using also the drone-based RGB also for disease.
Agre, can you please conclude?
Yes, I'm done.
Yeah.
So we have the IITA YAM Genome Hub that we recently developed. So this will allow you to search for any markers, any genotype, if it has been sequenced, which platform, and then retrieve all the markers information you need. So I think with that, I'm done. Thanks so much.
Thank you, Agre. So now that we have come to the end of our presentations, because we are a little bit short on time and I wouldn't want to keep our participants here for too long, we will give maybe 5 to 10 minutes of questions. I see there's already a hand up, so Ahmad, feel free to state your question and maybe also say who is it for out of all of the presenters. Thank you.
Thank you very much for allowing me to ask the question. Great presentations yet again. Another good day. Congratulations to all speakers. I asked first question from Agre. Okay. My question is, at what depth you sequenced your 332 genotypes for your whole genome resequencing? This was a diversity panel, and you were checking the diversity, right?
Yes, that was for diversity panel. So the depth was 30x.
30x. Okay.
Yes.
So at one point you mentioned that you identified 173 unique genotypes or accessions. Would you mind sharing the threshold that you put or to set to identify the uniqueness or identify the duplications? At what level of genetic distance? I mean, you said that, okay, they are the unique or they are the duplicates.
Yes. So the threshold or the genetic distance was 0.04. So those same genotype that we put in during the sequencing, we use them as a kind of threshold, and then those genotype come up to have a genetic distance of less or equals to 0.04. So for yam, that's what we use. I think for cassava, they went for 0.06, and then for maize, I think 0.04 also. So I think it's not really standard, but it doesn't vary much during that. So there's already a pipeline that I have already. I think is R package I can share with the committee so they can use it also. So you just have your sequencing information, then you plug in either the dosage information, then it give you all the information, and then you can use that one to select your single genotype.
Oh, okay. Okay, so there is no standard, but if we you have used a whole genome resequencing, right? And you put the threshold of 4%. That's
Yes.
correct?
Yes.
Okay. Okay. Thank you very much. I'm satisfied with the answers. Thank you, Peter. And I have another question from. I'm sorry, I forgot the name. The presenter before Agre.
Julie.
Yeah, Julie. Yeah, exactly. My question from Julie is she mentioned one software, GBS Dive. My question is, can we use this software for our whole genome resequencing data as well? As she mentioned about GBS and DART-seq, but my question is, can we use it for whole genome resequencing as well?
Hello, thank you for your question. Yes, you can. The only thing you need to have is a VCF file with your SNPs.
Oh, perfect.
So, and it should be more accurate because you should have a higher number of sites and a higher number of heterozygous sites. So the plots will be sharp and it will be easier to interpret.
Oh, perfect. Perfect. Thank you, Julie. That's it.
You're welcome.
Thank you, Ahmad, for your question and our presenters. We have another hand up from Elizabeth. Elizabeth, you have the floor.
Good morning again, colleagues. Good afternoon, good evening. Fantastic presentations all. I am echoing Ahmad's comments. Just one question, and this is a very practical one to Angre. Are you guys working on yam rust as well? Have you found any resistance or any varieties that are resistant to yam rust?
Yam rust, we may relate those one to nematode, what we call the dry rot or wet rot. We started a assemble different population last year. So we are yet to conclude the first analysis. So right now we don't have any. We have them physically in the field, but we have to test them and then confirm if what we are seeing is really a kind of like resistant. So we started already. So we have some population assembled and then we are phenotyping them. So probably this year or next year, we should be able to come up with kind of like a conclusion.
No, I'm sorry. I guess I didn't pronounce it right. Yam rust, like the leaves,
Can I
the
respond?
Okay.
Can I
Thank
respond to that, Agre? If you are
Yes,
okay with
please.
that?
Sure.
Yeah. So Elizabeth Johnson, that name actually echoes me. Where are you from?
Ranjana, we actually, I think, was on the panel. We were working on the banana TR4 group.
Yeah.
Yes.
Yes. So that name is so you know, and I'm
Familiar.
like, okay, I know her so much. Anyways, so I don't know where you are talking about the yam rust. I don't think we see it in West Africa. You know,
Really,
so
that's interesting.
yeah.
Because
So
we are now
mhm.
fighting with it in the Caribbean.
Oh, yeah, you are from the
So
Caribbean. Now I remember. Yeah.
yes,
Yes, but you know, it also you have to say whether it is in Alata you are seeing, because I guess it is in Alata, you know.
yes, it's a lot.
Yeah,
Yes,
because
for them
yeah,
in Caribbean is a lot.
yeah. What we see here is more like, you know, anthracnose disease, which is
Anthracnose.
coletotrichum, you know, glyrosporids. But maybe the rust one is also fungus, if I'm correct. Is it, which one
Yes,
is
it's
it?
a basidiomycete. So it you're
Uh-huh.
saying this is opportunistic from our region then.
I hope it remains there and doesn't come anywhere else. Yeah. So no, I don't think we do anything on rust so far. I don't think we have seen it here,
Okay.
but and I haven't seen our pathologists actually reporting about it. So I think it is contained in Caribbean for now, Elizabeth. So maybe, yeah, something that you can work on and then restrict it there, please, not in West Africa.
We'll try.
Thank you. But yeah, we should connect back, Elizabeth. Thank you so much.
Definitely.
Yes.
Thank you,
Yeah. Thank you.
Ranjana and Andre.
Thank you. Alessandra, I don't see any other hands up.
Yeah, no, I think we are good for today.
Yeah.
Thanks
I
so
think
much
so.
to
Yeah.
everyone, especially our speakers. And I mean, the presentations will be shared afterwards. So I think you will be able to ask more questions to our speakers if you want in writing. So I don't know, Ranjana, if you want to say anything else, I'll leave you the floor and then we can close.
No, I think we are all good. I think either the polyploid session was too oh, sorry, somebody, yeah, please. Somebody wanted to speak?
Yes, yes. Sorry. If we have a couple of minutes, then may I ask another question, just for a clarity?
Please go ahead.
Yeah, thank you very much. The question is for Agre. I want to know how to classify some of the genotypes if they are not clustering in some clear clusters or groups. For example, when we are preparing the dendrograms or when we are preparing principal component analysis figures, if some accessions are not clearly clustering or grouping with some groups, then how to describe those genotypes?
Yeah, we have a two option to do that one. You can set your threshold if you use a probably admission or if you use admission or boost trapping. So with the admission, you can declare your threshold from 60%, 70% up to 80%, depending on the crop you are working on. Then based on that, if the one that fell into a group, you classify them properly. The one that you fail to assign a group for them, you can classify them as admins. So we have what we call admins. That is the first option. The second option, what I was saying, that you can use the bootstrapping. So the bootstrapping with the level of the threshold also will help you to classify them properly. And then also those ones that fell under each group, now you can declare them also as admins or non-assigned. So you need to assign the threshold. So with the threshold now, the output results, you can use that one to assign each individual to each group.
Perfect. What is the other way? What is the threshold? Bootstrapping, and the other ways?
Admission.
Okay.
Yeah, so under the admission, you have the BIC, the Bayesian information criteria. It will help you also to clarify those information.
Perfect. Thank you, Agre. Thank you so much.
You're welcome.
Thank you, colleagues.
Yeah, I think with that, I think the polyploidy, I guess, was either very simple for everybody or it was too complex. But anyways, these are the crops that we live with and we eat them and we work with. So please the people now. It's a group that is small still around the world, but we are all working very hard and we are really trying to bring together this community for polyploids. And I really encourage like scientists like Dr. Elizabeth Johnson and many others that we have worked earlier, but now somehow we have again disconnected. But we should come together as a polyploid group and develop proposals which are cross country, cross region, cross geographies, and we are facing the same issues, and we submit to some potential donors and get funding and work together and learn from each other. With that note, thank you, Alessandra, for this fantastic topic, and I think we did justice to this to some extent.
Absolutely, more than justice.
and great, and please do reach out to us and we will work together. Some of you, I'm getting there. Dr. Elizabeth Johnson, you will get an email from me, surely. Thank you.
Fantastic. I look forward to it, Ranjana. It'll be great to connect again.
Thank you. Thank you very much. Please send me your email on this platform. Thank you.
Will do.
Have a great day, everyone.
Thank you. Bye-bye.
So much.
Thank you, Alessandra, for being a great.
Yeah.
Thank you, Agre. Thank you, Brigitte. Thank you, Julie. Bye-bye. Have a lovely day.
Yeah.
Bye.
Bye-bye.
Thank you.
Bye.